Hello students, welcome to GDC. In the today's video, we will talk about the video solution of the MCQs of microbiology. So let's come to the question number one. So first question is, who is father of microbiology? Let's understand what is microbiology. So microbiology is a branch of biology where we study about microorganisms. So we study about microorganisms. So what are the microorganisms? Microorganisms like your bacteria, virus, fungus, or fungi. We study about them. So your options are Louis Pasteur, Robert Hooke, A.V. Leeuwenhoek, and Robert Koch. So your right answer is option number C, A.V. Leeuwenhoek. So let's see the explanation. So Leeuwenhoek is universally acknowledged as the father of microbiology, and he discovered the both protista and bacteria. Remember, protist and bacteria both are discovered by your A.V. Leeuwenhoek. This is one of the important questions. So this is also associated with they formulate the gene germ therapy. They invented pasteurization. What is pasteurization technique? So in this pasteurization, we basically heat the milk to avoid the microorganism growth in that milk. Disease produced by microbes. They give the concept. Okay. Next question number two. So question number two is smallpox vaccine was discovered by. So who discovered? Your options are Robert Koch, Louis Pasteur, Robert Hooke, and your option number D is Edward Jenner. So who discovered smallpox vaccine? So your correct answer is your Edward Jenner. What the correct answer is Edward Jenner. Let's see the explanation. So smallpox vaccine was discovered by Edward Jenner in year 1796. That was the first successful vaccine that was discovered. Remember, that was the first successful vaccine that was to be developed. He observed the milkmaids who previously had caught cowpox did not catch the smallpox. So that inoculated vaccine are protected against inoculated variola virus. Okay, so first vaccine was developed by Edward Jenner. So name of that vaccine is your smallpox vaccine, and in the year 1796. Okay, next question. So next question is the term microbiology. Who coined the term microbiology? So your options are Leeuwenhoek, A.V. Leeuwenhoek, Carl Linnaeus, Louis Pasteur, and Robert Koch. So here the right option is your Louis Pasteur. So remember, he coined the term microbiology. Let's learn about this scientist. So Robert Hooke, who studied the section and saw the matrix teeny cylindrical-like structure called cell. So he discovered the cell. He published a famous book called Micrographia, who was sketch of various natural things under microscope. So in the Micrographia group, what he observed lively they draw in a book. Then Carl Linnaeus, they first universal classification of system of living beings. So living beings, Carl Linnaeus, what they do? So they basically classified the living beings in living beings. He created the system named plant and animal. First times they had developed a system that is plant and animals. It is also known as father of modern taxonomy. This is also important. Who is the father of modern taxonomy? You can also get a question like this. So remember, Carl Linnaeus is regarded as the father of modern taxonomy. Then Anthony von Leeuwenhoek. He was the published a letter by Royal Society in London that described animalcules. So he also called the bacteria as a animalcules or little animals. Okay, he is known as the father of microscopy and microbiology. He is the father of microscopy and microbiology. Okay, only microbes not described by Evi Liebenberg were virus. So in Evi Liebenberg did not describe any information about viruses. Next is Louis Pasteur. Louis Pasteur, he proposed that microbes were responsible for the illness. What he proposed? So there he proposed that microbes are in any disease, so microbes have roles for the development of that particular disease. Result the controversy of spontaneous generation of virus biogenesis. He discovered pasteurization and fermentation process, which will be discussed. He also discovered vaccine, that is rabies and anthrax. So remember, Louis Pasteur is associated with vaccine rabies and anthrax. So you can remember like Mr. Lara, a mnemonic, that is Mr. Lara. Okay. M for microbes, L for Louis Pasteur, A for anthrax, and R for rabies. Okay, you have to remember. Next question. Next is Robert Koch. Robert Koch gave the first demonstration of role of bacteria causing disease. This is important. So Louis Pasteur at the level of give the information first time about bacteria, but Robert Koch gave the first demonstration of role of bacteria in causing disease. So what is the role of bacteria in development of disease? First, he isolate the anthrax bacillus, that is Bacillus anthracis, that causes anthrax disease. Introduced to use the solid culture media. He introduced the solid culture media in 1882. In the year 1882, he discovered Microbacterium tuberculosis. This is very important question from the exam point of view. Remember, in 1882, what discovered? Mycobacterium tuberculosis. By who discovered? By Robert Koch. Robert Koch discovered Mycobacterium tuberculosis. And remember, what is this responsible? This Mycobacterium, this is responsible for the disease tuberculosis. Also, with Louis Pasteur, they both scientists gave the germ cell theory of disease, germ theory of disease. So, जो like first germ cell is associated with which scientist? That is, remember, Louis Pasteur and Robert Koch, two important scientists. That is Louis Pasteur and Robert Koch. They both gave the germ theory of disease. Koch four postulate microorganism fulfilling the criteria responsible for any disease. They also given by Robert Koch. Koch four postulates. Okay. Next question. Next question is gram positive bacteria contains typically contains. So next question is gram positive bacteria. What they typically contain? Cell wall that lack of peptidoglycan, repeating units of arbinogalactone and microlates in their cell wall, peptidoglycan, muramic acid and D-amino acid in their cell wall, and cell wall containing predominantly polysaccharides and glycoprotein. So you have four options. The gram-positive bacteria typically contains what? So your right option is option number C. That is gram-positive bacteria typically contains peptidoglycan, muramic acid and D-amino acid in their cell walls. Okay, if we'll see, so we divided my bacteria into two type, that is gram positive and gram negative. So gram positive bacteria cell wall is thick, that is 20 to 80 nanometer, whereas gram negative bacteria having cell wall is thin, that is 1.5 to 10 nanometer. Gram positive bacteria having higher peptidoglycan content. Remember, gram positive bacteria having high peptidoglycan content, whereas gram negative bacteria having lower peptidoglycan content. They are having lower peptidoglycan content. Low lipid content, so gram positive bacteria having low lipid content, whereas gram negative bacteria having high lipid content. So this difference is important from the exam point of view. Remember, gram positive cells, so how you remember? Gram positive cell having a thicker cell wall, gram negative having thinner cell wall. Gram positive cell is thick wall due to high peptidoglycan content, gram negative cell wall is thin due to low peptidoglycan content. If we talk about lipid content, so gram positive bacteria having low lipid content, gram negative bacterial cell wall is having high lipid content. The main component of gram positive bacteria is peptidoglycan, teichoic acid, trichronic acid, diuronic acid, muramic acid, and D-amino acid. Whereas the gram-negative bacterial cell wall contains peptidoglycan. Major is your lipopolysaccharide. Lipopolysaccharide is major, and lipoprotein. Remember, lipopolysaccharide basically present in the outer membrane of the gram-negative bacteria. Next is explanation of gram-positive bacteria is gram-positive bacteria show purple color while staining. So when gram-positive bacteria, remember positive, P for positive, P for purple. So gram-positive bacteria basically show purple color, whereas gram-negative bacteria basically show pink or red color while staining. This is you have to remember. Okay, so this is negative, not positive. This is negative. Okay, so what we learned? So gram-positive bacteria having thick cell wall, high protein content, low lipid content. Major component is peptidoglycan, teichoic acid, diuronic acid, muramic acid, and D-amino acid. Whereas gram-negative bacteria cell wall contains is thin, lower peptidyl content, higher lipid content. Major component that is present are lipoprotein, lipopolysaccharide, and so negative, a pink color, pink or red color. Next question. A Next question is a given gram positive bacteria is differentiated from gram negative bacteria by gram staining. This is because, so the question is gram positive bacteria and gram negative bacteria, they are basically different from each other by gram staining. So basically stains which part of the cell wall? Remember, gram stain basically stain your, or options are lysozymes, teichoic acid, membrane protein, a lipid. So your right option is option B, that is teichoic acid. Remember, in gram staining, the cell wall contains teichoic acid, which is stained by the staining, gram staining. The peptidoglycan layer of many gram-positive bacteria as densely functionalized with amino glycomers called as teichoic acid. So gram-negative bacteria is surrounded by three peptidoglycan, which is itself is surrounded by another outer layer, that is your lipopolysaccharide. You can see gram-positive bacteria having teichoic acid, which is stained by the gram staining, whereas gram-negative bacteria contains lipopolysaccharide instead of teichoic acid. Okay, so remember this is one important question. Which part is basically stained? Due to this, the teichoic acid is responsible for the staining of gram-positive bacteria. This is very important from exam point of view. Next question is which of the following combination are correct? So it has four statements. You have to tell which combination is correct and which is not. So first statement is it is saying that outer membrane absent in gram-negative bacteria. Gram negative bacteria is more resistant than the gram positive bacteria. Teichoic acid is found in gram positive bacteria. Gram positive bacteria is more permeable than gram negative bacteria. Okay, so your options are P and R are true, Q and S are false, P and S are true. Let's forget about the option. Let's learn what the statement is saying. Is this which one is correct or which one is not? Question first statement is outer membrane absent in gram negative bacteria. You will come to this staining. You will come to this part. You can see the outer membrane here. You can see in gram positive bacteria. You can see a outer membrane. You are not finding any gram outer membrane layer in gram negative bacteria. See, there is no outer layer. So first statement is correct. So outer membrane is absent in gram negative bacteria. That is correct. Next statement is gram negative bacteria is more resistant than the gram positive bacteria. This is correct. Remember, gram negative bacteria are more resistant as comparison to gram positive bacteria. Third is teichoic acids are found in gram positive bacteria. This is also correct. Next is gram positive bacteria is more permeability than no. So here the right option is QRS. Sorry, outer membrane is absent in gram negative. This one is false. Okay, this option is false. So gram positive bacteria is more permeable than the gram negative bacteria. Yes, your QRS option is correct. This is correct, and this one is your false. Your option D is your correct answer. Okay, next. So characteristic, you will see the comparison between gram positive and gram negative bacteria. So if we talk about the number of layer, so gram positive having one layer, whereas gram negative having two layers. If we talk about thickness, so as I told you, gram positive bacteria having thicker cell wall, gram negative bacteria having thinner cell wall. If we'll talk about outer membrane, remember gram positive bacteria contain outer membrane and gram negative bacteria, gram positive bacteria absence of outer membrane, gram negative bacteria having outer membrane. Periplasmic cell. So periplasmic space is present in some cell. Gram negative bacteria present in all the gram negative bacterial cell. Chemical cell wall. So what are the chemical composition of the cell wall of gram positive bacteria? That is your peptidoglycan, teichoic acid and lipoteichoic acid. Whereas gram negative bacteria's measure is your lipopolysaccharide. Protein are absent in gram positive bacteria. Protein are present in gram negative bacteria. Lipid are less in gram positive bacteria. Lipids are more in gram negative bacteria. Permeability is more in case of gram positive bacteria. Permeability is less in case of gram negative bacteria. Resistance is less and resistance is more. This table is very, very important from the exam point of view. So you have to remember that the basic information about gram positive and gram negative bacteria. So gram positive bacteria having thicker cell wall, gram negative bacteria having thinner cell wall. If we talk about the composition, gram positive bacteria contains gram positive bacteria cell wall contains peptidoglycan, teichoic acid, and lipoic acid, whereas gram negative bacteria contains liposaccharides. So like that, you have to remember all the point that has been discovered over here. So this is very, very important from the exam point of view. Next question. Next question is you have to tell the correct sequence of gram staining procedure. So you have to tell the correct sequence of gram staining procedure. First one is crystal violet, crystal violet, decolorization, iodine treatment, primary stain. So you have to tell the procedure, correct procedure. Option B is D. First, you have to do decolorization, then crystal violet, then iodine treatment, then primary stain. Third option is primary stain, crystal violet, iodine treatment, decolorization. Fourth one is iodine treatment, crystal violet, primary stain, decolorization. Here, the right option is your option number C. First, you have to do primary staining, then with crystal violet, then with iodine, then with decolorization. See, option C is correct. Let's learn. Gram staining. So gram staining was first introduced by in the year 1884 by scientist Christian Gram. So what is the procedure? First primary dye you have to use, that is crystal violet and methynyl blue. Add mordants such as gram iodines. Iodine bound strongly to the primary dye and wait for 1 minute. Then you have to add a decolorizer like acetone or alcohol. Then you have to add counter stain, that is sulfurin. Observation of gram positive bacteria purple color, gram negative bacteria pink or red color. The gram staining is used for the distinguish. Remember, this gram staining is basically used for the distinguish between gram positive and gram negative bacteria. Okay. So gram positive, first you have to do fixation. Fixation के बाद you have to do crystal violet, then iodine treatment, then decolorization, then counter stain with sulfurin. If it is purple color, then it is gram positive. If it is either pink or red color, it is gram negative. This is very, very important from the exam point of view. In the year 1884, this staining was first discovered by Christian Gram. Okay. Next question. Next question is Petri plate was distinguished for solid culture media by. So who discovered Petri plate, which is used for the solid culture media? Your options are Robert Koch, Robert Petri, Richard Petri, Alexander Petri. So here the right option is your Richard Petri. Okay, option number C is the correct answer. So the Petri dish was developed by German physician Julius Richard Petri, after whom the name given as Petri plate. So we called it as a Petri plate while working as an assistant to Robert Koch at Berlin University. Okay. Next question. Next question is a specimen obtained from a patient's cerebrospinal fluid, cultured in a specialized media for about 5 weeks, show the presence of a bent rods and tested with gel Nissel reagent. Identify the gram organism. This question was asked in GPAT 2001. So your question is when a cerebrospinal fluid of a particular patient has been obtained and when it is cultured for the 5 weeks, she show the presence of bent rod and tested positive of gel initial. Remember, gel Nelson staining is associated for Mycobacterium tuberculosis. This is very, very important question. Why Mycobacterium tuberculosis, that is your tuberculosis causing microorganism. So test positive test with 𝘎𝙬 reagent. So if we talk about 𝘎𝙬 staining, or we called it as a acid fast staining. So we called it as a acid fast staining. So reagents are primary dye as a carbon fusin. Sometimes question also asked which is the primary dye used for the 𝘎𝙬 staining. That is your carbachol fusin. Then carbon decolorization as 20% sulfuric acid, counter stain methynyl blue acid fast bacteria give a red or to purple due to the return of carbon to give color. If it is acid fast, bright red to intensive purple. If it is non-acid fast, it will give blue color. So acid fast stains are basically used for the differentiate fast organisms such as Mycobacterium tuberculosis. Okay, high content of mycolic acid in their cell wall basically responsible for this staining. So remember, the question can be asked, which is the major component that is present in the acid fast microorganism to give a positive to acid fast staining or 𝘎𝙬 staining? That is your mycolic acid. That is basically nothing. That is what it is, a mycolic acid. This is important from the exam point of view. Okay. Let's go to the next question. Next question is saying that a specimen is isolated from a patient suffering from septicemia and was found to be strict aerobic. Okay, this is a aerobic microorganism. Its culture vial had a characteristic group-like order and was susceptible to carbonicillin. So that culture media having a order, and this is susceptible to carbonicillin. Okay, that is penicillin sensitive. Okay, what is this microorganism? So options are Pseudomonas fluorescens, Salmonella typhi, Staphylococcus aureus, or Pseudomonas aeruginosa. Here the right option will be Pseudomonas aeruginosa. Okay. So see carbonicillin is active against Pseudomonas. Carbonicillin is a which class organism? This is a penicillin derivative. Penicillin. That is penicillin which class antibiotic? Beta lactam antibiotic. And this is organism is strict aerobic. This is active against aerobic microorganism and group like order causes septicemia. Pseudomonas fluorescens is a strict aerobic, but carbonicillin is not active against these three microbes. Pseudomonas is a aerobic, but this carbonicillin is ineffective against this Pseudomonas fluorescens. But Pseudomonas aeruginosa is a common capsulated. This is a gram negative. Remember, this is a gram negative. So you have to remember that your penicillin class of the drug. Penicillin is effective against gram negative microorganism. That's why later stage they develop broad spectrum antibiotic from the penicillin. Na? So this is effective in gram negative, aerobic, facultative, anaerobic, and rod shaped bacterium that causes disease in plant and animals, including humans. Okay, your right answer will be Pseudomonas aeruginosa. So remember, carbonicillin is effective against Pseudomonas aeruginosa. Pseudomonas aeruginosa is if responsible for the septicemia and it provides a specific odor. Okay, next mordant used in gram staining. Just I have discussed with few slide back, which is used as a mordant for gram staining. So your options are methylene blue, surfarin, grams iodine, acetone. The right answer is your grams iodine. So grams iodine is used as a mordant. Okay, this is very, very important. So let's see. Just I have discussed. So it was this training was discovered in year 1884 by scientist Christian Graham. So it first the primary dye is used is question can be asked about primary dye. Primary dye is your crystal violet or methylene blue. Mordant what are used such as grams iodine. Mordant used that is your grams iodine. Decolorizer what are the decolorizer used? Decolorizer like your acetone and alcohol. And observation, if it is gram positive, it will give purple color. If it is gram negative, it will give either red or pink color. Okay. So gram staining basically used for the distinguish between gram positive microorganism and gram negative microorganism. Okay. This question is just discussed. Next question is Hansen's disease is also called as what is the, that means what is the synonym of Hansen's disease? Leprosy, diphtheria, typhoid, and none of the above. So here the right answer is leprosy. Remember, leprosy is also known as Hansen's disease. So this is also this is occurs due to a microorganism that is Mycobacterium leprae. So this disease of the skin is this basically affect your skin, your peripheral nerves, your mucosal surface of the upper respiratory tract and the eyes. Leprosy is known to occur at all ages, ranging from the childhood to old. So you can see the fingers are diffused. You can see the fingers are diffused. This is a typical symptoms associated with leprosy. Okay, so this is the symptoms associated with leprosy. Leprosy is also known as Hansen's disease. This is caused by microorganism that is Mycobacterium leprae. So one important drug that is sulfonamide class of the drug that is dapsone. So that is primarily used for this case, leprosy treatment. Okay. Next question is my microscopic examination of the culture isolated from a spherical, revealed from a spherical bodies with a smooth outline of growing in long chains, identify the microorganism. Microscopic examination of culture. Okay, microscopic examination of a culture isolated revealed that the spherical bodies with a smooth outline growing in a long chain identify the microorganism. Your options are Staphylococcus aureus, Staphylococcus pyogenes, sorry, Rhizostops stolonifer, and Bacillus subtilis. This question was asked in year GPAN 2000. So your options is Staphylococcus aureus. The right option is your Staphylococcus aureus. So let's see, that is chain or cocci, that is these are spherical body, these are also called as streptococci. Okay, the culture is of staphylococcus, sorry, streptococcus pyogenes. This is a gram positive, spherical or ovoid, arranged in a long chain and non-motile. Staphylococcus aureus are gram positives, non-spore forming, facultative anaerobic, non-motile, catalysis positive or negative. They are present in a grape-like structure. Okay, they are basically present like this. Okay, they will be present like, see, this is the grape-like structure. Okay, they are also occurs in pairs and occasionally in short chains. You can see these occurs in pair, sometimes in short chains. Rhizostom stolonifera is a fungus. Bacillus subtilis is basically a gram positive. Okay, you have to remember the classification of microorganisms, bacteria. Okay, so then after you will able to answer this question. Next question is decolorizer used in gram staining. So just now I have discussed what is the decolorizer used. So your options are crystal violet, alcohol, sulfurin, yeah, all of these. Your right option is C. Decolorizer, what is used? That is your alcohol or acetone. Alcohol or acetone. Primary dye is used, that is crystal violet, which give purple color for gram positive and gram negative. Moderant, moderant is iodine color, both give purple color. Decolorizer is acetone and alcohol and acetone, both acetone for gram positivity give purple color, gram negative colorless. Counter stain that is responsible for the identification, sulfurin or carbon cosine. When you counter stain appear, if purple color is if present, this is a gram positive. If pink or red color present, that will be gram negative. This you have to remember. Okay. Next question. Next question is, which of the following organism is a acid fast microorganism? Which is acid fast microorganism? This is very important. Just I have discussed with you. Mycobacterium tuberculosis, E. coli, Bacillus subtilis, या Pseudomonas aeruginosa. Your right option will be Mycobacterium tuberculosis. This is a microorganism that is acid fast microorganism. Okay, so mycobacteria is called as acid fast because they group of rod shape that can be seen under microscope following staining procedure, that is bacteria retain color. So they basically identified by acid fast staining or gel nasal staining. Okay, acid fast staining and gel nasal staining. So bacillus subtilis, E. coli and Pseudomonas aeruginosa and Staphylococcus epidermis have live or dead fluorescence like staining. Okay, you can see such type of staining will be observed with Mycobacterium tuberculosis. Next question. Next question is a compound microscope. This is very, very important. Who discovered compound microscope? A.B. Leeuwenhoek, Louis Pasteur, Jamson and Hans, yeah, Robert Hooke. Your right option will be Jamson and Hans. So see, Hans and Jaster and Janssen in 1590. This is a dotty eyeglass marker invented first compound microscope tube with lens at each end. Robert Hooke in year 1635 to 1703. This is a chemist, mathematician, physicist, and innovator. Improves compound microscope. They improves the compound microscope. Anthony Leeuwenhoek. These invented simple microscope. Okay, compound. You have to remember who discovered simple microscope, who discovered compound microscope. Next question. Next question is acid fast microorgan acid fastness in acid fast staining is due to abundance of, that means which compound or chemical present in acid fast microorganism that is sensitive to, that basically responsible for the this staining, acid fast staining. Options are your mycolic acid, squalene, ergosterol, peptidoglycan. Here the right option will be mycolic acid. Okay, see, acid fastness of microbacteria is basically due to high amount of mycolic acid in their cell wall, due to high amount of mycolic acid in their cell walls, which is responsible for the staining pattern of poor absorption followed by high retention. Okay, this is cell wall, that is capsules, this is mesosomes, the plasma membrane, this is cytoplasm, DNA, and this is ribosome. This is a typical structure of a acid fast microorganism. Typical feature of acid fast microorganisms. Okay, let's move to the next question. Next question is presence of mitochondria in living cell can be determined by use. So for the determination of mitochondria, what you used for a living cells? Your options are neutral red, methylene blue, Janus green, yeah, phenolphthalein. So here the right option is Janus green. Okay, Janus green is used for the determination or identification of mitochondria. Okay, if you will see the structure of mitochondria, mitochondria having a matrix and that is a inner membrane. Mitochondria having DNA. Remember, mitochondria DNA is basically comes from mother, not from father. This is also one of the important question. Or maternal body, this is obtained from mother or maternal body. It has a outer membrane and inner membrane, and this is also contain ribosomes, and this finger like structure is called as cristae. So this is Janus green is a basic dye that is a vital stain used for in this histology. It is found the stain of mitochondrial supravitality. So they basically used to stain the mitochondria supravitality. Janus green B, that is JGB dye, is used for the vital staining of mitochondria and reduction and oxidation, so the electron transfer chain alteration. The defect in electron transfer chain of mitochondria by paraquat is linked to the free radical formation. Remember, your mitochondria is responsible for the oxidation reduction balance. Okay, so this your mitochondria. If we'll talk about your mitochondria, if we talk about your mitochondria basically do your oxidate balance between your oxidation and reduction. Any damage like a paraquat, any damage due to any agent that will cause redox imbalance, and this redox imbalance is associated with generation of free radical like your H2O2 O2 minus OH peroxynityl that can induce lipid peroxidation and oxidative stress. Lipid peroxidation and oxidative stress. Okay, this is due to damage of mitochondria. Next question, what is the region of decolorization in gram-negative bacilli in gram staining? So why we do decolorization step? So your options are due to teichoic acid, cell membrane, due to dyes used in staining, या lipid content. Here the right option is your decolorization agent. Okay, so decolorization agent like ethanol or acetone solution is interact with the lipid of the membrane. So they interact with the lipid in, interact with the lipids. Okay, so this decolorizer, the region for decolorization is your nothing but your lipid content. Okay, so option number D is correct answer. So lipids of the membrane of both gram positive and gram negative bacteria. The outer membrane of gram negative bacterial cell is lost from the cell having peptidoglycan layer. Okay, so gram positive, so due to the lipid content, so they basically decolorizing agent react with the lipid of. So you can see gram positive bacteria having peptidoglycan layer, periplasmic cells, gram negative bacteria having more periplasmic cell. Gram positive bacteria having more peptidoglycan, gram negative bacteria having less peptidoglycan. Gram negative bacteria having outer membrane, gram positive bacteria having no outer membrane. No outer membrane. Cytoplasmic cells, here is the cytoplasmic cell. So instead, in gram negative bacteria, extra agents are present that is called as your lipopolysaccharides. Okay, that is called as lipopolysaccharides. Okay, let's move to the next question. Next question is Albert's A solution consists of. Albert's A solution consists of what? Silver colloidal suspension of tannic acid, toluidine blue, malachite green, yeah, Felugen. So Albert A solution contain what? So your right option is toluidine blue. If we'll talk about Albert staining use two solution, that is Albert A and Albert B. Albert A solution consists of toluidine blue. Remember, Albert staining contain Albert A solution and Albert B solution. Albert A solution contains toluidine blue, glacial acetic acid, and alcohol. Toluidine blue, glacial acetic acid, and alcohol. That is 95% ethanol. Albert B solution contains iodine and potassium iodide. So result is when it reacts with cyanobacteria diphtheria is present in the sample, it appears as a green color and rod shaped bacteria is arranged at an angle to each other. Next is nucleic acid staining. In nucleic acid staining, your Felugen is used. For Albert staining, your Albert A, Albert B. Albert A contains toluidine blue, uh, toluidine blue, glacial acetic acid, and alcohol. So you can remember by a simple mnemonic. Albert staining is what are the reagent used by our exam GPAT. So G for G you have to remember glacial acetic acid. For P you have to remember, so you have to remember potassium iodide and iodide. A for alcohol and T for toluidine blue. So you can remember by simple mnemonic GPAT. Okay, at the Albert A and Albert B. Albert A contains GAT and Albert B contains P, that is potassium iodide and iodide. Next is nucleic acid staining. Nucleic acid staining, that is Felugen. Felugen is used as a identification of chromosomal material. Okay, Felugen is used for the identification of nucleic acid staining. That is means your chromosomal material or DNA in the cell specimen depending upon the hydrolysis of DNA. So this is basically the staining of the DNA bacteria. So bacteria DNA is basically stained by nucleic acid staining. Next is flagella staining. This flagella staining employs for the mordant silver colloidal suspension of tannic acid and stain basic fusin. This coat flagella with stain until they are thick and enough to be seen. Then determination of bacteria containing flagella. Okay, so this is for the determination of bacteria containing flagella structure. Next is scapular Fulton staining. This staining is used basically malachite green, and the counter stain is used, that is your sarparin. The bacterial endos, this basically stains your bacterial endospore. Okay, so let me summarize what we learned from this slide. So we learned four major type of staining for the bacteria. First one is your Albert staining. Second one is your nuclear staining. Third one is your flagellar staining. Fourth one is your Schaeffer's Fulton staining. So Albert staining having, as I told you, you can remember by simple mnemonic GPAD, G for glacial acetic acid, P for potassium iodide and iodide, and A for alcohol, that is 95%, T for toluene. Albert staining contains two type of solution, Albert A and Albert B. Albert A contains solid in blue, glacial acetic acid and alcohol. Albert B contains potassium iodide and iodide. So what they basically result, they used for the identification of cyanobacterium diphtheria. Next is nucleic acid staining. This is basically used for the staining of DNA in bacteria. They basically use Phelugen. That phelugen basically stains the chromosomal material of the DNA. Next, in flagellar staining, this flagellar staining is employed as a moderant of silver colloidal suspension of tannic acid and stain basic fusin. Next is Schaeffer's Fulton staining, that is basically used as a primary stain of malachite green and counter stain as a sarfarin. This is basically stain your bacterial endosome. Okay, next question number 21. So next question is simple staining is used for. Why this simple staining is basically used? Your options are study the cellular organelles, revealing basic cell shape, differentiating two different type of cells, or understanding the working of mitochondria. Here the correct answer is option number B, revealing the basic cell shape. So if we'll talk about simple staining is you perform only for one basic stain. Okay, that is your either crystal violet or methylene blue or basic fusion. This is principle of simple staining. All the bacteria in a smear takes stain and appears to be color of stain. Basic stains are more affinity toward the bacteria surface and stains of the bacteria. It is used for the study of morphology and arrangement and shape of the bacteria. What they basically give information of bacteria, they give morphology information, they give how this bacteria is arranged, and they will give information about the shape of the bacteria. What is the basic shape of a bacteria? Then procedure, if we talk about the procedure, a bacteria smear is prepared, air dried and heat fixed. This heat fixed smear is folded with either one of the basic stain and allowed to react with 1 to 2 minutes and then wash under running tap water. Now it is air dried and focused with 10x, 45x and 100x. So this simple staining is basically performed for three major information: for the study of morphology of the bacteria, study the arrangement, how the pattern of arrangement of bacteria. Third one is shape of a bacteria. So if we talk about the procedure, first bacteria, you have to prepare the bacterial smear. Now you have to air dried and fix with methanol or heat, sorry, fix with heat. So when you give fixed, then this bacteria will be completely fixed over this microscopic slide. Now this heat fixed smear is flooded with either one or basic stain. Okay, now it can be again further dry to view under microscope with 10x, 45x and 100x staining. Okay, next is endotoxin. So endotoxin is produced by gram-negative bacteria, is present in peptidoglycan, lipopolysaccharide, teichoic acid, inner membrane. Okay, so remember, so your right answer is your lipoic acid. So remember what is endotoxin. So endotoxin is basically obtained by gram-negative bacteria, and these endotoxins are pyrogenic. Pyrogenic means if the endotoxin is enter into the bacteria, that will induce fever. That's why whenever we prepare any parenteral formulation, whenever we prepare any parenteral formulation, so we have always go for measurement of endotoxin. So endotoxin are the lipopolysaccharide found in the cell wall of gram negative bacteria, they which induces basically inflammation and fever as a immune response in higher organism. Inflammation and fever. Lipopolysaccharides are large molecule. These are basically consist of lipids and polysaccharide composed of O antigen and outer core and inner core, and they are joined together by a covalent bond. They are found in the outer membrane of gram negative bacteria. They are basically found in outer membrane of gram negative bacteria. So these lipopolysaccharides, the endotoxins are the lipopolysaccharide. They are basically gram negative present in gram negative bacteria, and they are responsible for the inducing inflammation and immune response in microorganism. I hope everyone is clear. Next question is the diiocinate of methylene blue belongs to which dye? So methylene blue basically comes under which dye? Acidic dye, basic dye, neutral dye, yeah, oxygen dye. Your right option is neutral dye. So stains or dyes. So based on the charge, so they are divided into either acidic or basic or neutral. Acidic के पास acidic have positive charge, basic have positive charge, acidic have negative charge, and neutrals having both charges. Okay, so if acidic dye containing basic dye containing positive charge, they produce colored cation with colorless anion. Example is methylene blue chloride. This is a basic dye, methylene blue chloride. And a negative dye or acidic dye example is eosin plus sodium. Neutral dye is eosinate of methylene blue. You have to remember the exam. This is very important from exam. You can get a direct which of the following is a neutral dye. You can also get a question like this. Okay, that's why you have to remember all these points very carefully. Next question is endospore. Endospore can be stained with. Options are malachite green, methylene blue, crystal violet, sarfarin. This is already I have discussed with you. It can be stained by malachite green. Okay, here the right option will be option number D, that is malachite green. Okay, so if we'll talk about this explanation, that is Kapfer-Fulton staining, or that is basically for the staining of bacterial endospore. So primary stain used, that is malachite green is used, primary stain, and the counter stain surfarin is used. It is used an alkaline substance. They basically used an alkaline substance that can stain the spore of the bacteria. This is used, malachite green is used for the staining of bacterial endospore. You can see the staining procedure. So first application of malachite green as a primary stain. Now application of heat for moderant. Now application of water for decolorization. Then application of sarfarin for the counter staining. Okay. Next, next question is which organism is identified by Albert staining? So which organism basically identified by Albert staining? This is also I have discussed in previous slides. So options are Cyanobacterium diphtheria, Clostridium botulinum, Salmonella typhi, none of the above. Here the right option will be Corynebacterium diphtheria. Okay, Albert staining. This is a type of differential staining used for the staining of high molecular weight polymer. Okay, this staining is basically used for the high molecular weight polymers of phosphate. High molecular weight polymers of polyphosphate known as metachromatic granules or Voltins granules found in the Corynebacterium diphtheria. Now these metachromatic granules are also found in Yersinia pestis and Mycobacterium species. Okay, so you can join this course by applying GPAT code GDC GPAT 2024 for the appearing mock test. This you can download this app from the Google or Play Store. Okay, that in the name GDC classes. Okay, this is the you can participate more and more number of questions, and here you can see your all India rank. Okay, that's all India rank, and you can see what is your where you are standing in all over the India. Okay, thank you. In the next part, we will discuss the next rest of the MCQs. Thank you.